Hello, everyone, and welcome to today's keynote webinar titled Exosomes versus Liposomes as Next Generation Therapeutics. Today's webinar is part of the 8th annual event in the Drug Discovery and Development Virtual Event Series. I'm Christina Jewell of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots. Now, before we begin, I would like to remind everyone that today's event is interactive, and we encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. And finally, as a reminder, today's presentation is educational and it does offer free continuing education credits. Click on the continuing education credits image located at the right of your screen and follow the process to obtain your credits. Now let's get started. I now present today's speaker, Dr. Volkmar Weissig, Professor of Pharmacology and Biomedical Sciences, Midwestern University. For a complete biography on our speaker, please find it on the left of your screen. Dr. Weissig, welcome. You may now begin your presentation.
Well, first, I would like to thank very much for the invitation, for this opportunity. And I'm very happy to be here. So I would like to compare exosomes with liposomes. By the way, I call them liposomes. Other people call them liposomes. That's still open for debate. And before that, I need to declare some conflict of interest. I have been working with liposomes since 1981. And I even did my second Ph.D., or in Germany, it's the habilitation about liposomes in 1991. I have been for many years the associate editor of the Journal of Liposome Research. And since 2021, I've been editor-in-chief. What I'm trying to say is, in this comparison, I certainly am a little bit biased when it comes to liposomes. Let's look at a very brief history of exosomes. For the first time, they have been observed in 1877, but nobody knew what it was, and it was called blood dust. They have been kind of rediscovered in 1975 when so-called microvesicles have been found in calf serum. In 1981, the term exosomes was coined for such vesicles between 50 and roughly 1,000 nanometer in diameter. Very important, at the end of the 1990s, at the end of the last century, the role of exosomes in cell-to-cell communication was revealed, and I'm coming a little bit back to it. And finally, after 2000, I can't put it in any other way, this field of exosomes or extracellular vesicles has literally exploded. In 2011, the International Society for Extracellular Vesicles was founded. Since then, a large number of so-called position papers, statements have been published, and I don't go into the details of these papers listed on this slide. I just go into the last one, the most recent one, which was published in December 2023, so-called MISEF or MISEF 2023, Minimal Information for Studies of Extracellular Vesicles from Basic to Advanced Approaches. And in these guidelines, or in this paper, extracellular particles are divided into vesicular and non-vesicular particles. And vesicular particles, in turn, are subdivided into extracellular vesicles, synthetic vesicles, and artificial cell-derived vesicles. And in the following, I will go into the definition of these three types of vesicular vesicles. Extracellular vesicles, very general, are all vesicles which in one or the other way are released from cells. They are delimited by a lipid bilayer And of course, they are not living, they cannot replicate. So everything the cell is releasing and which has a lipid bilayer around it is an extracellular vesicle. An artificial cell-derived vesicle involves vesicles which are produced when we disrupt cells, when we lyse them or when we tonicate them, and what's left are then artificial cell-derived vesicles. Synthetic vesicles, per definition from this MySEF 2023, are synthesized de novo from molecular components. And here I have already an issue as a liposome guy, because vesicles synthesized de novo from molecular components, and of course we have to talk about phospholipids, have been known already since the beginning of the 1960s, when Alex Bengham discovered these vesicles. And these vesicles, actually, people try to call these vesicles bangasomes, according to Alex Bengham, but he declined it. So, and since 1968, these vesicles are known as liposomes. But they are now, according to this definition, synthetic extracellular vesicles. and oops, sorry. Sorry again, have to get used to this. And another type of synthetic vesicles are created when extracellular vesicles or native extracellular vesicles are being fused with liposomes. Now, that's the guideline from this last statement. Other authors are using different classifications. Like in this paper, they distinguish between exosomes and microvesicles. And this is based on the biogenesis. Exosomes are derived from endosomes, and microvesicles are produced by or are derived from plasma membranes. And in the next slide, I show more details about this mechanism, how these two types of vesicles are being generated. Exosomes are released after fusion of endosome-derived multivesicular bodies with a plasma membrane. And I have highlighted this. So early endosomes, they turn into multivesicular bodies, and they then fuse with membranes, with plasma membrane, and they are being released. So extremely important, exosomes are derived from endosomes. In contrast, microvesicles, they are released just by direct budding of a plasma membrane. So, and that's a very important feature which distinguishes exosomes from microvesicles. So what are the major features of extracellular vesicles? First of all, they are a natural delivery system for a large variety of different cargo. As you can see in this cartoon, all enzymes, RNAs, peptides, and chaperones, proteins, they can be transported, they can be delivered via extracellular vesicles. Depending on the source where these extracellular vesicles are coming from, we can traverse certain biological membranes. They are biocompatible, of course, and also depending on the source, they display a certain tropism towards specific organs or tissues. They have a low immunogenicity, and of course, they are non-living entities. The major physiological or cell biological functions of extracellular vesicles are first, as I mentioned already, cell-to-cell signaling, discovered in 1996. Immune regulation. They play a role for tissue remodeling. They play a role for waste disposal. And they also play a role for development and differentiation of cells. And last but not least, diseases or the progression of diseases can be affected if the mechanism to produce extracellular vesicles and to circulate extracellular vesicles are malfunctioning. I would like to highlight again the function of extracellular vesicles for cell-to-cell signaling, and this was nicely summarized in this quote, and I'm only reading the red part. Extracellular vesicles have revolutionized the way we view the communication circuitry in humans and between the kingdoms of life. So how does it work? Very schematically, cells can release diverse extracellular vesicle subtypes. And here we have microvesicles, exosomes, and we also have microsomes and mid-body remnants. The last ones are not included in the classification systems I introduced earlier. Well, they are being released by the cell. We get to neighboring cells, and we can, depending on the cargo we deliver to these cells, and upon uptake by these cells, we can elicit local signaling response. And we can also enter the circulation, and then we can go anywhere in the body, and we can elicit systemic signaling responses. Some examples, what extracellular vesicles actually can deliver, what kind of functional cargo we can deliver. First, we can deliver signaling proteins. We can deliver messenger RNAs. We can deliver microRNAs. And we can deliver long-coding RNAs. In other words, we can deliver a lot of signals which in the end will affect the transcription and translation in the receiving cell. So in very important messenger systems. And obviously, just looking at these examples, it certainly opens up whole new avenues for future therapies. And that's nicely summarized in the next quote from 2024. Researchers are now fully galvanized in harnessing extracellular vesicles as cell-free therapeutics to treat various diseases. And by the way, I would like to mention now, I use the term extracellular vesicles and exosomes interchangeable. So I don't really distinguish. So I will refer to exosomes or to EVs throughout my talk. So currently, there are over 300 EV-based clinical trials underway. And if you look at this diagram, the most of it, 50% are dealing with biomarkers, and 28% dealing with using exosomes for therapy, and other areas are analysis, drug delivery systems, which I come back to, and cancer vaccines. Let's look at some more details here. It means I give you some examples for the use of extracellular vesicles as biomarkers. It has been found that exosomal microRNA in lung cancer patients is different from healthy controls, so very important biomarker. It has been found that urinary exosomes contain tumor-specific protein markers, which is very important for diagnosis, for early diagnosis of prostate cancer. It has been found that different microRNAs are differently expressed or have different levels in exosomes derived from the cerebrospinal fluid in Alzheimer's patients. And the same applies to cardiovascular diseases. We have found a different expression profile of microRNAs in circulating exosomes of patients with myocardial infarction and heart failure. So these are now some examples for the potential use of exosomes as biomarkers. That means for the early diagnosis of any kind of disease. Now let's look at some examples for using of exosomes for therapies. Wound healing. It has been found that exosomes derived from mesenchymal stem cells promote tissue repair and regeneration. So actually, that means they accelerate the wound healing process. Exosomes loaded with interleukin 12 can elicit tumor-specific immune responses. And they also have been found to have an anti-inflammatory effect. That means they can modulate the immune response and reduce inflammation. The market for exosomes is big, as you can see on this global market analysis. And if you look at this, I mean, the curve actually starts going up kind of exponentially already. So a big market is out there, and that makes a lot of sense. And I quote now from this market analysis. In the next slide, I take a quote from this particular market analysis. It says, the exploration of exosome therapeutics represents a significant advancement in biomedical research offering promising avenues for targeted drug delivery and regenerative medicine. So I highlighted in red promising avenues for targeted drug delivery because that's the aspect of exosomes I would like to focus on. So now let's look at exosomes or extracellular vesicles as a drug delivery system. And I showed you earlier, approximately 5% of the currently 300 plus clinical trials are dealing with this. So exosomes as drug delivery systems. And by this, I mean not the delivery of endogenous cargo. This involves the use of exosomes and loading them with an endogenous drug and then using it as a drug delivery system. Well, exosomes are being praised, highly praised. In this paper, they are being celebrated as a rising star for drug delivery. Pictures like this are populating the literature. Exosomes as a drug delivery system. You can see that exosomes can deliver pretty much everything to anywhere in the body. Great drug delivery system, supposedly. In the guidelines, which I just introduced already, it has been stated exosomes are more, first, it has been stated that more effective and safe carriers are needed. No argument about this. Then they write, EVs are a potential solution. With a natural lipid bilayer and with a lack of immunogenicity and lower toxicity compared to lipid nanoparticles. And I have a big issue with that. First, they compare it to lipid nanoparticles. Lipid nanoparticles contain cationic lipids, which are non-physiological, non-natural lipids, and they have some toxicity. But these lipid nanoparticles are essentially used only for the delivery of RNA. Just let's look at the COVID vaccines. So that means EVs actually should be compared to liposomes and not to lipid nanoparticles. And the next paper, even more, some of the vital features of exosomes, such as biocompatibility, low toxicity, and low immunogenicity, make it a more exciting drug delivery system. Again, that's the claim raised by the exosome community. Recently, or last year, click chemistry was highly praised as a new tool to modify the surface of exosomes. So that means by modifying the surface of exosomes means you take an exosome and you add additional ligand targeting moieties to the surface of exosomes. And by doing so, you supposedly can make exosomes target specific. And I quote from this paper, this approach holds great promise for personalized and effective cancer treatment, paving the way for next generation therapeutics. Well, when I actually became aware, again, I'm a liposomologist, and when I became aware of this hype around exosomes as a drug delivery system, I started to pay attention to this field. And that's why I think I'm giving this talk right now. Because considering this hype about exosomes as a great drug delivery system, I just would like to hit the pause button. Why? First, a biocompatible, non-toxic, and non-immunogenic drug delivery system has been introduced already 50 plus years ago. Gregoriadis described in 1971 liposomes as carriers of enzymes or drugs and as a new approach for treatment of storage diseases. In 1973, he discusses in detail the drug entrapment in liposomes. And that's shown in this cartoon. It's just a normal liposome. I don't have a pointer here. In blue, the phospholipids, and different colors, hydrophobic or hydrophilic drug molecules. And it shows that liposomes can encapsulate both types of cargo, hydrophilic or hydrophobic. And he, in 1974, these are three classic papers, actually, three landmark papers. Gregoriadis discusses a drug carrier potential of liposomes in cancer chemotherapy at the beginning of the 1970s. Further, let's look at the surface modification. the development of liposomes with surface-linked targeting ligands began 45 plus years ago, Laserman described the binding of antigen-bearing liposomes to specific tumor cells. And he also describes the targeting of liposomes which have been covalently coupled with monoclonal antibodies or protein A to certain cells. So targeting of these liposomes. There is no shortage of conjugation methods. And actually, if I may say, I myself contributed to this field in 1986. We described a new hydrophobic anchor for the attachment of proteins to liposomal membranes. This was 3 years later commercialized by Avanti Polar Lipids. Actually, I could not profit from this because we did this in East Europe and we didn't have sufficient money to file for patent protection in West Europe, in America, but doesn't matter now. And click chemistry, as I showed earlier, praised as a new tool for modification of exosomes, has already been applied to liposomes in 2006. Pictures like this, functionalized liposomes, have been populating the liposome literature for the last 40 years. If you look at this picture, you can attach immune globulin, peptides, aptamers, etc., to the surface of liposomes. That's great. And when this idea was developed in the early 1980s, I mean, it caused a lot of euphoria because pharmaceutical scientists were hoping that finally the big dream from Paul Ehrlich about the magic bullet becomes reality. So liposomes were thought to become the magic bullet. We just put a surface, a targeting moiety on the surface of liposomes, and protein, peptide, carbohydrate, and then we can direct these liposomes anywhere in the cell. So again, this was developed, these ideas were developed in the 1980s. Now, where are we today? Today, there are more than 20 liposomal drugs in the clinic. And I don't go into the details here, but it's also too small a print. The thing I would like to highlight is the next slide. Oops, I always press next here. I got to put next slide. 20 liposomal drugs in the clinic, but there is not a single FDA-approved drug in the clinic which is based on liposomes with surface-linked targeting ligands. So not a single liposome is in the clinic which has on the surface some kind of peptides or proteins in order to render it cell-specific. And this is a very important point, which I really would like to tell to the exosome community, because that's certainly something to keep in mind. Now, why? It's a big problem, and I don't have a clear answer. I could only imagine two different aspects. First, if you look at this previous slide, this surface modification, if you look at this slide again, to produce such complicated, complex system pharmaceutically on a large scale might be challenging, number one. And the next aspect is liposomes actually can act as an adjuvant. I mean, I myself was working in Gregoriadis' laboratory in the early 1990s, and we covalently coupled, using my method, peptides to the surface of these liposomes, and we could see that we have triggered immune response. So that means as soon as you add something to the surface of liposomes, you might trigger an immune response. Well, why should exosomes be different? So now, what were the major hurdles and problems liposome technology had to overcome in order to bring liposomes into the clinic? First, it was the lack of regulatory guidance. The first guidance for the industry on liposomal drug production, and including chemistry, manufacturing, controls, etc., was issued in 2002. This was 7 years after the first liposomal drug, Doxil, which I introduced shortly, was FDA approved. And these guidelines were updated most recently, not most recently, just in 2018. And I would like to state now that if exosomes, which should be used for drug delivery, would have to meet the same criteria liposomes have to meet according to these guidelines, Well, I would say exosomes, we would not have a chance to get FDA approved. So subsequently, new guidelines for the translation of exosomes into the clinic need to be developed, and that's exactly what people are doing in 2023, guidelines for clinical translation and commercialization of extracellular vesicles and exosome-based therapeutics. So what are major problems, or what is the most striking difference between exosomes and liposomes? Of course, as you can see here, the degree of complexity. Exosomes are derived from endosomes or from budding of cell membranes. So that means we can contain a large variety of cargo, shown here. And this cartoon doesn't even refer to the high variability of the phospholipid composition, which also adds to it, depending on where the exosome it has been derived from. And if you compare this with a liposome, and this is doxyl, you see doxyl in the middle, the red stuff is precipitated doxorubicin. Getting back to it, you have a phospholipid membrane surrounding it, and defined phospholipid membrane with a known lipid composition, and you also have polyethylene glycol on the surface. So, and if you compare the complexity, you can easily imagine that this complexity might become or is a big issue for large-scale production and for regulation. So according to the guidelines, we correctly state that the complex structure of EVs is a double-edged sword. Why? First, on one hand, I mean, the good thing is these EVs, natural therapeutic content, sure. And we can have also targeting proteins from parent cells, depending on where they are coming from. That means we are able to diverse to cross specific cell membranes. So that's a good thing. But on the other hand, as I mentioned already earlier, the heterogeneity of EVs makes the purification process, release criteria, difficult to standardize. And this paper about the guidelines states, and I quote, "In this regard, the FDA needs to be convinced that such heterogeneity is an inherent biological feature of EVs with likely advantages, and now most important, there is no real need to overcome it in a therapeutic product." But again, this is still work in progress. Oops. So now coming to doxyl, what are the specific lessons which we have learned from the development of doxyls? Okay. The first doxyl, as I explained already earlier, is liposomal encapsulated doxorubicin. Has been FDA approved in 1995. This essentially was the first nano drug delivery system worldwide approved. It was indicated first for AIDS-related Kaposi sarcoma and then extended to ovarian cancer and multiple myeloma. The production, the development of doxyl is based on 3 different unrelated principles. First, prolonged drug circulation time and avoidance of the RES system. Second, high and stable remote loading of doxorubicin, driven by a new method, by a so-called transmembrane ammonium sulfate gradient. And third, the lipid bilayer of doxyl is in a so-called liquid ordered phase. That means we have a high phase temperature transition. So again, 3 aspects had to be addressed. And I think these are aspects also individuals, companies want to develop exosomes have to address. The half-life of circulation, the concentration of a drug inside the vesicle, and of course the drug leakage from the vesicle. So how did we, and I wasn't personally involved, but how did the liposome community solve these 3 problems? Let's start with the half-life in circulation. Already in 1972, Gregoriadis has injected radioactively labeled liposomes into rats, and he found that the bulk of liposomal radioactivity is removed from the blood within minutes. So extremely short circulation time. useless if you want to use liposome as a drug delivery systems in humans. Now, certainly no, I mean, this problem was solved with the pegylation. That means polyethylene glycol chains were attached to the surface of liposomes, and this cartoon is a classic drawing from Dan Lasik. On the left-hand side, you see a so-called naked liposomes, and on the right-hand side, you see liposomes with polyethylene glycol chain, and this schematically illustrates that these polyethylene glycol chains prevent the interaction of liposomal membrane with opsonines, so making it stable in circulation. As a result, the circulation half-life of doxyl went up to 90 hours, and this was sufficient in order to utilize the enhanced permeability retention effect. It means in order to develop doxyl as an anti-cancer drug. And if you have questions, I can go more into this, but I mean, not right now. However, the commonly view that nanocarriers, liposomes, conjugated to polyethylene glycol are non-immunogenic has been proven wrong. This paper, pegylated liposomes, immunological responses. And remember, I said earlier that anything which is on the surface of liposomes, I refer to peptides, can become immunogenic. Now, why would it be different for polyethylene glycol? Polyethylene glycol itself is non-immunogenic. It's being used in many different applications. But once you attach it to vesicles, we can produce antibodies. And that's the first effect. It has been found that polyethylene glycol linked to the liposomes promotes the formation of antibodies. And this results in the ABC effect, the so-called accelerated blood clearance phenomena. That means if you re-inject such liposomes, the half-life in circulation is drastically reduced. And the second effect, and by the way, I would like to mention here, I mean, COVID vaccines, they contain pegylated phospholipids, and it has been found that COVID vaccines can induce the formation of antibodies. And that's a different topic. So that means, okay. And it has been found that a polyethylene glycol linked to the surface of liposomes also can activate the complement system, and that's is reflected in the so-called in a complement activation release pseudoallergy. So now that's one problem with polyethylene glycol. But there's another problem, the targeting effect. Pegylation interferes with active targeting. And this is very simply shown in this cartoon. This is a naked liposome, only with antibodies on the surface. It certainly is target specific, I mean in vitro, but in vivo you wouldn't be able to use it because it's being rapidly cleared. Now, if we introduce polyethylene glycol, as shown here, we certainly have a reduced clearance. That means we have a longer half-life, but obviously polyethylene glycol will interfere with the targeting effect. It blocks the antibodies. So the way out of this dilemma was to link the targeting moiety to the distal end of polyethylene glycol, but then you expose again a protein to the environment, and that means it has been found that the circulation time of such constructs is inversely proportional to the density of ligands bound to the distal end of PEG. So I'm emphasizing this because extracellular vesicles, exosomes, as the well-praised next-generation drug delivery platform, will face the same problems liposomes did more than 30 years ago. And if you look at this cartoon, post-modified EVs, and in the middle you see EV-inspired liposomes. That's a long stretch. I mean, we have shown such liposome pictures, drawn such liposome pictures long before EVs have been known. So, I mean, we have not been inspired by the EVs. But anyway, I want to emphasize just separating the targeting moiety, the antibody, from the polyethylene glycol we also want to use. This doesn't really cut it. Okay, so it's not possible. So in other words, at this point, I don't see any advantage exosomes would bring in comparison to liposomes. But will keep going. Now, since polyethylene glycol obviously, turned out to be immunogenic, toxic, and there is literature about the toxicity of COVID vaccines based on this phenomena. You can look it up. Other options to extend the half-life of exosomes, which are being studied according to this paper from 2021, are to attach some proteins. Now, CD47 is the so-called don't eat me signal. That means it prevents the uptake by macrophages and other cells. CD31, BECAM1, essentially the same, and CD24 molecules which promote the adherence of vesicles to other cells. And beta-2 macroglobulin is a part of a major histocompatibility complex. That means it's involved in the process of recognizing self versus non-self. So that means there are certainly approaches worth being pursued in order to avoid the problems with polyethylene glycol. These methods, these proteins, these approaches have not yet applied to liposomes, but I would like to remind you there might be a reason why proteins linked to liposomes haven't made it into the clinic. So again, exosomes will face the same problems. Now let's go to the concentration of the encapsulated drug. Regarding the potency of this drug, it has been calculated that the concentration of the drug inside the liposomes has to be in the millimolar range. And for this, a new loading method had to be developed, and that's the so-called remote loading via a transmembrane ammonium ion gradient. If you look at the circle, again, unfortunately, I don't have a pointer function here. If you look at the circle, we encapsulate ammonium sulfate. Ammonium sulfate dissociates, and ammonia diffuses out of the vesicle. As a result, the interior of a liposome becomes acidic. And doxorubicin, on the right side, you see it, is a weak base. And in the non-charged form, it can diffuse into the liposome, becomes protonated, and it precipitates. So it's like a sink condition. That means it's being doxorubicin diffuses in, precipitates, the equilibrium keeps being shifted to the left. And by doing so, they were able to encapsulate up to 90% of the doxorubicin, or what is more important, the concentration inside the liposome is about more than 100-fold the drug level in the loading medium. So that's a big thing. That means you have liposomes, and the drug inside the liposome, the concentration is 100 times higher than the drug in the loading medium. Now, whether any of these methods, which are praised by exosomologists, can achieve the same high drug encapsulation efficiency remains to be seen. But I can only say all of these methods have been tried to load doxorubicin into liposomes, and they did not work. So why would it be different with exosomes? I don't know. Now, finally, the third point is once the drug is inside the liposome or the exosome, it has to stay inside. And this cartoon shows a leakage kinetic study of doxil. On the left panel, you see in red the doxorubicin crystal, and you see the leakage is a two-step process. First, the crystal has to dissolve, sure, and then doxorubicin can passively diffuse through the membrane. Shown here, shown here. And this is the part I want to focus. That means the release of drugs from phospholipid vesicles, be it a liposome, be it an exosome, through the phospholipid membrane. What does it depend upon? Of course, the physical-chemical properties of a drug are extremely important for right balance between hydrophobicity and hydrophilicity. But there's nothing we can do about this when we have a certain drug. It's a given we cannot change the physical-chemical properties of the drug we want to develop. The concentration gradient obviously is important. I mean, the higher the concentration gradient, the higher is the tendency to leak out. And there's also nothing we can do about it because we want to have a high concentration gradient because we want to encapsulate a lot of drug. This leaves the lipid composition, and I marked it in red because that's the only thing we actually can influence. So what effect does the lipid composition and the phospholipid composition of a vesicle, be it exosome or liposome, has on drug retention? Phospholipid membranes exist in different phases depending on the temperature. This panel on the left-hand side, you see the gel phase, and just let's skip the rippled phase. At the main transition temperature, then it switches into the liquid phase. So the rippled phase is just in transition, not so important for now. So either gel phase or liquid phase, and this depends on the temperature. Now, the point is now that the liquid phase, as you can maybe schematically see on this cartoon on the right-hand side, this liquid phase has a higher non-specific permeability. So the gel phase is tighter, it's more difficult for a drug to diffuse out. Liquid phase, the drug can easily escape from the vesicle interior. This is a thermogram of doxyl. In red, doxyl, and in black, empty doxyl liposomes. That means the same liposomes used for encapsulation doxyl, just without doxyl, and doxorubicin. And you can see the phase transition temperature is around 54 degrees centigrade. In other words, the membrane of doxyl at body temperature exists in the gel phase, which has a lower non-specific permeability. And why is that so? For the generation of doxyl, we have used hydrogenated phosphatidylcholine from soybeans. And natural soybean, and natural PC from soybean, you see at the bottom, has a high content of unsaturated fatty acids, oleic acid, linoleic acid. And if you make phospholipid membranes of natural soy PC with a high amount of unsaturated fatty acids, you have a transition temperature far below the freezing point. Once you hydrogenate it, it jumps up to 55. So this was the trick we have used to stably incorporate doxorubicin into liposomes. Now the question, of course, is now to what extent this method could be used for exosomes, whether we can hydrogenate lipids and exosome membranes. Well, it might not work. Of course, I would also emphasize, this is only a problem if the drug retention emerges as a problem, because if the drug, for example, is extremely hydrophilic, then the tendency to get out of the vesicle is lower. But again, the phase transition temperature of a membrane here also plays a role. We are in time, 42 minutes, okay. So finally, as final conclusions, I would like to summarize what are the major challenges for translation of EVs into the clinic. Some of them I addressed already, some of them I haven't addressed yet. First, all issues associated with isolation, production, and purification. The lack of standardized protocols, that's something that can be overcome. The complexity of isolation of pure population, that's an issue, I come back to it a little bit. Very important, for the production of exosomes, we have to use natural sources, and there's certainly a low yield of exosomes from natural sources. Don't know how to scale it up. We can also co-isolate contaminants, and very, very important is also the batch-to-batch variability. It was a big issue for the development of doxyl. It has to be proven that each batch is the same. And I see issues and problems when these criteria are being applied to exosomes. Next, characterization and quantification. I emphasized already the heterogeneity of EVs and also the detection and resolution of subtle differences in subpopulations. And here, actually, I just 3 hours ago, I found an advertisement on LinkedIn from a company, and they asked the same question. How do you characterize the heterogeneity in EVs? How do you identify subpopulations within EV samples? How do you assess the molecular composition of EVs? And how do you perform all of this at a single vesicle level? Well, and this company, I don't give you the name because I'm not getting paid by this company for advertising them. They have a method, it's called single molecule localization microscopy. which might be able to overcome this problem in terms of quantification and characterization of exosomes. Further, cargo loading, I have addressed this already. I mean, depending on the potential, on the potency of the drug, you might have to have a high concentration of a drug inside the vesicle. If you have a highly potent drug, then maybe it's not a problem. This drug loading has to be reproducible. And I showed you earlier this slide, which shows the different loading methods which are being investigated for loading exosomes. And the question, of course, is these methods certainly will affect the biological function of exosomes. The cargo variability, exosomes are derived from parent cells. So that means depending on the physiological or pathological state of the parent cells, exosomes will be different each time the parent cell changes. So I mean, exercise, medications can influence the cargo of exosomes. I address the target and delivery issue. Again, I mean, and big advantage of exosomes is if we are derived from vesicles and we are from cells and they are a mean of communicating. So of course we are able to penetrate or to cross the membrane of a receiving cell. But this is something different versus using them as a universal drug delivery system. And then you have to make them able to pass the barrier of other phospholipid membranes, maybe of other cells which you want to target. Further, immunogenicity and safety. I mean, we can be immunogenic, again, depending on the source. Toxicity, EVs from cancer cells may carry pro-inflammatory factors, or we can also facilitate tumor growth and metastasis. All of this depends, again, on the parent cell. And I have not mentioned stability and storage issues with liposomes, which also was a problem, the shelf life. I mean, you can liposomes store for 2 or 3 weeks and then become to deteriorate. So that's not sufficient. So a lot of work went into developing methods to cryopreserve them in the presence of trehalose. You can freeze-fry them, and you can then sell them, deliver them as a lyophilizate, and then you can reconstitute it. And of course, the stability in biological fluids is also an issue which relates to the half-life in circulation. And finally, I mean, regulatory and manufacturing challenges. The lack of standardized protocols can be overcome. The cost of manufacturing is also an important thing. I mean, I mentioned earlier, I was trying to address the question, why is there no liposome in the clinic with surface-linked whatever ligands? I mean, and one aspect is certainly also it might be too expensive to large-scale manufacture such complex systems. But then the last point is the regulatory uncertainty, but that's certainly something which will be overcome. So that means, I remind you what I said, the FDA has to be convinced that the complexity is not a disadvantage, and it should not be an issue for the development of exosomes. So I'm pretty much at the end now. I would like to thank you very much for your attention. And as a final message, maybe, I would like to state something which I expressed also earlier in other places. I think that exosomologists first should pay attention to lectures learned 40, 50 plus years ago in the area of liposomology. And in other words, I would like to encourage exosomologists to work together with liposomologists in order to get exosomes as a drug delivery system into the clinic. So again, I thank you very much for your attention, and please feel free to contact me at any time using this email address. Thank you.
Dr. Weissig, thank you so much for your informative presentation. It is now time for our live Q&A portion of our webinar. So to our audience, if you haven't already done so, or if you have and you have some more questions, please submit them now. Just type them into the Ask a Question box, click Submit, and we will try to get through all of our questions. So Dr. Weissig, we have quite a few questions that have already come in. I'm just going to dive right in and we'll see how many we can get through. So our first one, can autologous EVs resolve the issue of immunogenicity and increase half-life?
Yeah. Well, I would say no. Or yes and no. I mean, because we have to do the same what we did in the liposomes. I mean, the immunogenicity. I mean, again, we have to distinguish between exosomes. I mean, let me say it differently. I'm only focusing on the use of exosomes as a drug delivery systems for drugs which have not been in the exosome in the first place. And to make them target specific, we have to get targeted ligands and then become immunogenic, just like protein-bearing liposomes. And what was the other question?
And can it increase half-life?
Okay, yeah. Yes, if we pegylate exosomes, we can increase the half-life, but we will face the same problems liposomes have been facing and are facing. And we are trying to use different methods by attaching these different proteins on the surface. This might also help to increase the half-life, but again, this makes these proteins immunogenic. So it's kind of currently, again, as soon as it was observed that also COVID vaccines, due to the back content, can be a little bit harmful, and I have to be careful in saying this here so out loud, but they can be immunogenic. I mean, everything you put on the surface of a vesicle eventually will become immunogenic. That means will trigger immune response. And in general, I don't know how this will be overcome. I mean, you can attach endogenous molecules, you can attach to the surface of vesicles, and by doing so, you present them to the immune system and they produce antibodies. So, I mean, again, I mean, after 40 years in development, there is not a single liposome in the clinic which has something on the surface except protein glycol. And what a surprise, even this doesn't really work. So I'm a little bit pessimistic here.
Well, we appreciate your honest answer. We have some great questions coming in, and this is a good one. Thank you, doctor. Are there any other commonly purported solutions to liposome cargo loading that you are skeptical of?
No, I mean, if I go back to this, I mean, can I quickly go back here?
You can.
As soon as possible. Oops. This one. Okay, these are classic methods. Well, skeptical. I mean, let's start with co-incubation. Number one. I mean, if you incubate exosomes with a drug in solution, then depending on the concentration of a drug in solution, some of the drug might diffuse into the vesicle, but there will be no gradient. So the concentration is very low, but again, if you have a highly potent drug, it might work. Now, sonication, well, good luck. You certainly destroy everything with sonication. Electroporation, you make a big hole in the exosomes, and you actually will remove the stuff inside the exosomes. I mean, freeze-thawing cycle might work. Extrusion, actually not. Transfection is a different issue. And saponin, we all have issues, okay? And if I would like to say is, you have to, I mean, whoever wants to develop something, it strictly depends on the properties of a drug and on the properties of a vesicle. If you have exosomes, you don't have much choice. You have to use the exosome you want to use. And if you have liposomes, you have a big choice in terms of choosing the right phospholipids. And then you have to optimize loading process in order to have a high loading capacity, in order to achieve a high loading capacity. And again, with Doxil, and I can say this, I'm not sure about transfection now, but all of these methods have been tried to encapsulate any kind of drug into liposomes during the 80s and 90s. In the 80s, it's really a big hype. Give me a drug and I will encapsulate it in liposomes. And let's see what happens, okay? So all these methods have been tested, but none of these methods have achieved such a high drug concentration in the vesicle as we need for doxorubicin. And that's why we have come up with this remote loading procedure, which can be varied. I mean, this was developed for a weak acid, for a weak base. Doxorubicin is a weak base. It can be turned around for loading a weak acid. So there's a lot of variability which can be used. So I'm not sure if I have answered this question. I mean, there is no universal method available. And if you are in the laboratory, if you are a student, you just have to try them out.
Thank you. Next question. Is there a way to manipulate the formation of EVs in vivo?
Yeah, yes and no. I mean, and there is a nice, yes. I mean, there is a nice work being done by, I forgot her name now. I mean, for example, exosomes have been used recently to deliver mitochondria into other cells. You know, mitochondria, the powerhouse of a cell, and many more things. And in order to enrich exosomes derived from cells with mitochondria, we have exposed these cells to conditions which promote mitochondrial biogenesis. So we have exposed cells to different things, which is a different issue now. As a result, cells became enriched in mitochondria, and as a result, for exosomes, or in this case, it was vesicles, budding membrane, we also had a higher concentration of mitochondria. So it means one can manipulate the host cell in order to increase the concentration of a certain cargo in the exosome. In general, that's possible.
Thank you. Next question. How does one stimulate EV production of a cell without causing any bias, such as changes in the cargo?
Yeah, that's actually a question which you should ask an exosomologist. Okay, I cannot answer this. I made it very clear. I'm a strong liposomologist and mitochondriologist, and I cannot answer this question. I'm sorry.
No problem. So this one is a detailed question. So bear with me here. Thank you a lot for your comprehensive presentation. I would like to ask a question about encapsulation efficiency while working with liposomes. So when utilizing liposomes as a carrier system, it is essential to assess encapsulation efficiency at various stages of the process. However, due to the structural properties of liposomes, assays such as BCA, Bradford may yield unreliable results as lipid components that can interfere with the reagents. Are there any alternative methods that offer a more rapid and reliable solution?
So that means the point is you use any kind of method to put something into the liposomes, and then you want to see how much is in it. And to do so, I mean, the first step is always you have to separate the liposomes from the drug or whatever which has not been encapsulated. I mean, that's obvious. Yeah, and then, I mean, it really depends on the cargo, on the drug. I mean, you have to find an assay which is highly sensitive, which detects the molecule you have put into the liposome. And there are all different assays available. And I went through it myself, and it's tough, okay? And in the end, it also comes to in order to detect what's inside, how much is inside, usually you have to lyse the liposome in order to release the cargo. And then the crucial point is these different methods to lyse them, for example, using detergents, they may interfere with the assay you are going to use. So all the controls of the assay, first you have to test whether the detergent you are using to lyse it interferes. If you don't want to lyse it, maybe there is a way to detect the compound inside the liposomes, but then maybe there are methods which you don't have to release the load from the liposomes. But again, I mean, there is no universal answer to this. It really depends on the particular drug you are looking at. So if you are a student working on this, I would first start looking for all methods which would be suitable to detect specifically this particular drug. That's number one. And it goes from simple absorption assays to HPLC and others. Yeah, and then check, I mean, which one of them is being interfered by the presence of phospholipids, because the phospholipids you have always there. So it's a tough task for young students. I know that, again, and I learned it the hard way. But there is only one way to go forward. You have to try all these different methods. But I mean, I don't know who was asking this question. I mean, I'm more than open. You have my email address. and I'm more than open to discuss more details. I mean, I've been working for liposomes for 40 plus years, and maybe I can give some advice once I know what particular component, what particular drug you are discussing. So I offer my help of free consultation if you would like to.
Thank you. And I took control of the slides just to get your email address right out in front of everybody. And I know we're almost at time. Do you have time to squeeze in a few more questions?
Sure, I have time, yes.
Excellent. I'd like to just add a couple more in here for our audience. Great presentation. What are the barriers to use the cargo of extracellular vesicles to diagnose infections?
Wow. Again, that's a question for an exosomologist. I don't know, but I only could speculate that whatever infection you have, I mean, and now I start thinking out loud, I mean, the cell is infected with whatever, and this certainly should be reflected in the exosomes. So that means now, I mean, to find out, I would use an infected, sounds funny now, infected individual, whatever, and to isolate exosomes from serum, and then I would try to detect, for example, a viral load in the serum. So that means whatever has affected the organism, the tissue, the cell, certainly eventually will end up in exosomes, and then this could be used as a biomarker. So that means, for example, just I introduce one example, that a prostate-specific antigens have been found in urinary exosomes. That's the same thing. I mean, it's and that means even maybe in urinary exosomes you can detect some viral particle or whatever infection you might have. I mean, and I'm pretty sure, I mean, somebody is doing this. I mean, again, the literature is exploding. So, I mean, I highly recommend going to PubMed and put search terms, exosomes, viral infection. And I'm sure there are papers out there who are dealing with this already.
Dr. Weissig, I want to thank you so much for this Q&A session. I'm going to wrap with this final question. Many thanks for your presentation. My question is about the aggregate formation due to lipid structure. For example, exosomes tend to aggregate during freeze-thaw cycles. Do liposomes also tend to aggregate? And if so, how has this situation been overcome for its transition to clinic as drug delivery systems?
Yes. If you have charged liposomes, if you have charged neutral liposomes, they certainly tend to aggregate. That's why if you put them in the refrigerator, after 1 week or so, they sediment, and you can sometimes resuspend them, but by resuspending them, you have to put energy into it, and by doing so, you can make them leaky. That's a problem. Now, if you have charged liposomes, I mean, negatively charged liposomes, they tend to be more stable because of repulsion of electrostatic forces. It makes sense. And exosomes might be also negatively charged, I think. I don't know, but they are natural sources, and cell membranes essentially are negatively charged. So I would assume that extracellular vesicles derived from cells via membrane budding are also negatively charged. So they should be a little more stable. However, if you freeze them, yes, then you certainly support the aggregation process, and that's actually that's why there is one method to drug loading is freeze thawing. You freeze liposomes in the presence of a free drug. You thaw them, you freeze, you thaw, and by continuously doing so, you disturb the integrity of a membrane, and so the drug can get inside the vesicle. You can do this with exosomes, I think. And then how it has been overcome, I mean, well, to be honest, I don't even know now. Maybe I should know this. I said more than 20 liposomal drug products are in the clinic, but it's a good question. I don't know whether any of these drug products are being supplied to the clinic in a lyophilized form. I don't know. I'm ashamed now. But I do know that you can lyophilize liposomes, and you can resuspend them. And to what extent? I mean, and then the results can be twofold. First, I mean, it can become leaky, and depending on the physical chemical properties of the drug inside, you will lose the encapsulated drug, number one. And then you also might change the size distribution. That's another important issue, especially for regulatory for purpose of regulation. I mean, these liposome products, they had to be very homogeneous. The size distribution has to be very tightly controlled and homogeneous. And when you freeze for them, I mean, the size distribution changes and become more heterogeneous. This can hurt the application, but not necessarily so. It also depends on the particular application you are looking into. I don't know if I've answered this question, but again, if not, I mean, you have my email.
Dr. Weissig, thank you so much for sharing your experience and your knowledge with the world here. Do you have any final comments for our audience?
Me? Yeah, I mean, I don't know how many liposomologists are among you and how many exosomologists are among you. And if you are an exosomologist, go into the literature of liposome technology. There are many, many books, and you will find a lot of information. I have edited myself a lot of books also. I mean, and you will be surprised what issues we had already addressed and had to solve, which you are now facing as a young exosomologist. Again, I emphasize, if you are an exosomologist who wants to use exosomes as a drug delivery system. I'm not talking about the other applications of exosomes. Big issue, okay? So, and I encourage, you know, sometimes I have a feeling, I mean, also the posts I see on social media, etc. Sometimes I have a feeling, I shouldn't say this, that young scientists dealing with exosomes, they don't even really know what liposomes are. And they are kind of, when I'm looking at these publications, which I have also referenced in my presentation, I have a feeling sometimes we are trying to invent the wheel a second time. So my advice is, look at the liposome literature. You can learn a lot if you want to use exosomes as a drug delivery system. And if you are a liposomologist, be open-minded, look in the exosome literature, and you can also learn from them, okay? Because exosomes certainly have a new potential, absolutely. I didn't mean to downgrade them in any way. So that means in the end, again, I highly encourage both camps, liposomologists and exosomologists, if I may use the term, to work tightly together.
Excellent advice from an expert. Well, Dr. Weissig, thank you again so much for sharing your time today and your important research. Before we go, I do want to thank our audience for joining us as well and for their interesting questions. We had so many great questions coming in. I'm sorry we went over, but I wanted to make sure we got these into our recordings for those who come back and watch it on demand. And speaking of on demand, today's webcast can be viewed on demand for 1 year, and that's till February 19th, 2026. LabRoots will alert you via email when this webinar is available for replay, and we encourage you to share it with your colleagues and your friends who may have missed today's live event. Dr. Weissig, thank you again so much for joining us, and to our audience, thank you again. Have a beautiful day, everyone. Bye-bye.
Thank you very much.